Linkage and gene mapping. In this session, we are going to discuss about the linkage. Linkage is an exception to Mendelian dihybrid inheritance. So the very objective of this session is to know about the linkage and also how to do the gene mapping. So first of all, what exactly the linkage is? We know very well that genes are linearly placed on a chromosome, right? And when the genes are very close to each other, they have a tendency to inherit together. So the tendency of the gene to inherit together is actually called as a linkage. And we can say that the haploid number of any species constitute a linkage group. For instance, in case of human beings, you see the 2n number is 46. So altogether we have a 23 linkage group. Bacteria has a single chromosome, right? Single circular chromosome. That's why we have a single linkage group. Okay. As far as linkage is concerned, first of all, it was reported by Bateson and Punnett when they were trying to repeat the experiment of Mendel, but for a change they took sweet pea instead of garden pea. And later on, the explanation for the linkage was provided by Morgan. So we will discuss both experiments, Bateson and Punnett as well as the Morgan, right? So first of all, have a look at Bateson and Punnett experiment. As I told you that Bateson and Punnett was doing the experiment of Mendel, and for a change they have taken sweet pea instead of garden pea. So they took two characters, one the long pollen shape and the flower color being purple. Okay, so long was dominant over round and purple was dominant over red. Okay, so when the two plants were crossed, then in the F1 generation we got purple long as per the Mendelian concept, because the law of dominance is operating. So in the F2, what happens? They got the four phenotype like purple long, purple round, and red long, and they were expecting as per the Mendelian ratio, that is 9:3:1. But for a surprise, they could not get that ratio. Instead of that, they found that the parental combination were far more. So purple long was 7, so was the red round, and the number of recombinants were very less. So that was the observation what Bateson and Punnett found while doing experiment with the sweet pea. Okay. Later on, they tried to explain the reason but could not explain exactly what exactly the reason. Okay. So they observed that Mendel's law of independent assortment is not operating. Parental forms are much more than the recombinants and the number of recombinants are very less. So as I told you, they failed to give explanation that why their result deviated from Mendel's observation. Later on, what happens? Morgan did the experiment on Drosophila and he explained the phenomena of the linkage. And on the basis of Morgan experiment, later it was concluded that gene for flower color and pollen shape in case of sweet pea was linked. So that is why the parental combinations were much more than the recombinants one. So in order to understand, first let's understand the Morgan experiment. Now what Morgan did, Morgan studied two different cross. Okay. So and he involved two traits, one long wing, that was a wild. Wild means that was a natural and the dominant one, and this is the mutant over the recessive gene forming short wings. And the another character was grey body, that is also wild and natural and is dominant over the recessive black one. So he took two characters, the wing and the color of the body. Okay. Now both the genes were placed on the second chromosome of Drosophila, right? So have a look at experiment number 1. So what he did, long wing and grey bodies were crossed with a short wing and black bodies. So you can see there, now it is placed on a chromosome, the genes are shown over there, the plus is denoting the wild form, okay, and these are the complete recessive form. So in the F1 we get this gametes, and finally the long wing grey bodies, this were the F1 phenotype, that is very much on the expected line. Okay. So this was the F1 genotype, right, that is the complete heterozygous. Now the test cross was done. Okay. Now in case of test cross, what happens that the F1 was taken, that is a long wing grey bodies were taken and they were crossed with a double recessive parent. Okay. And in the F1 what they observed that the long wing grey bodies were much more, that is 41%, so were the case of short wing black bodies, right, and the recombinants were just 9%. Okay. So again it was a deviation from Mendel's law, because had the Mendel's law been operating, then all should have been 25% each, because we know very well that in case of Mendel the test cross is going to give a phenotypic ratio of 1 1 1 1. So again it is a deviating from that, right. Now before we conclude, let's see the Morgan's experiment number 2. So in the experiment number 2 he took long wing and black bodies. Mind it, this was a dominant one, this was a recessive one. Here also, so this was a recessive one and this is a dominant one. Okay, like this you can see there. So here the parental will become long wing black bodies and here short wing grey bodies. So in F1 again we are going to get the same thing, that is long wing grey bodies, because long is dominant over short and grey is dominant over black. So that's why in F1 as per the law of dominance we are getting long wing grey bodies. Okay. So this was the genotype. Mind it, in the previous case the genotypes, in the previous case the genes were, the dominant genes were placed on the same chromosome and the recessive on the same chromosome, but here it is just reversed. So that is the difference you can see there. Later on we will see the very concept of cis and trans. So what happens as a test cross, the parental combination now long wing black bodies, this now we get 41%, short wing grey body, this now we get 41%, and the recombinants as per the new cross, the long wing grey bodies and the short wing black bodies, now you are getting only 9%. So again the parental combinations were much more. That was the observation. It is the same observation what we have seen in the experiment number 1 as well. So on that particular basis Morgan observed that in cross 1 F1 carries the wild form on the same homology, and this is actually called as a coupling or cis configuration. So this is present on the same chromosome, that's why we are calling them coupling or cis configuration. And in F2 it was on other chromosome, one dominant, one recessive. This is what we call repulsion or trans configurations. Okay. So what was the conclusion? The conclusion was, irrespective of the fact whether cross was done in cis or trans, F1 produced gametes. Percentage of recombinants in both cases remain the same, and that is less, and parental combination are much more. So it can be easily concluded that genes for body color and wing shape are linked. So that's why they inherit together. Since they inherit together, so that's why the parental combinations are more. So this is the thing what we observed in case of linkage. Now there is one illustrative problem. A test cross of F1 flies produces the following offsprings. So on that particular basis, what we can say, that heterozygotes are either in cis configuration or trans configurations. It is very clear that plus A, that means one dominant, one recessive, okay, they have a more combination, that's why it is going to be in trans configurations. Had it been the case of plus and here we have got 41%, then we could have said that it is in cis configuration. So by looking at the genotypic constitution, we can say whether the given example is cis or trans configuration, right? Now let's talk about the linkage, the types of linkage. There are two types of linkage, one what we call complete and second is the incomplete linkage. In case of complete linkage, there will be no recombinants, only parental will forms, but in case of incomplete linkage, recombinants are formed and their number will be less. Here no crossing over, but here crossing over happening. Okay, and here what we see, parental forms are much more than the recombinants. So this is the difference between complete linkage and incomplete linkage. Now first of all, if you talk about the complete linkage, you see, so this is the complete linkage I have taken in the form of a chromosome, the different alleles are shown over there, since there will be no crossing over, so what will happen? The gametes will be like that. So this one will be constituted over here and another, this one is going to be constituted over here. So a perfect heterozygote is going to be formed. So under the meiosis, what will happen? The gametes is going to be formed like this only because there is no crossing over. Okay. So what we see, that since A and B are very close to each other, so there is no possibility of crossing over in between them, right? So only parental forms are formed, recombinants are not formed, and usually these are very, very rare in occurrence, right? So this is about the complete linkage. Now let's talk about the incomplete linkage. If suppose A and B, there is a sufficient distance between A and B that some sort of crossing over is going to happen, then what will you see? That it is going to have a crossover, and as a result of crossover, what will happen? That 4 different possible are formed: AB, then a small ab, then capital AB, and finally small a, capital B. So these are the possibilities that is going to be formed, right? So what we see, 80% parental form, 20% recombinants, right? Because we have taken into consideration 20% recombinants. So what we see, here A and B are not close to each other, so that's why crossing over between them is possible. Parental types are more, less recombinants, and this is of frequent occurrence in our nature, right? So if we want to differentiate between incomplete linkage and independent assortment, we can show like that, up till this point there is no difference, right? That here also this is going to form heterozygotes, okay? But in the next, what we see, that since A and B are linked, so what will happen? The parental forms will be more and recombinants will be less. But if we talk about independent assortment, what will happen? Equal proportion of all type of gametes are formed, okay? Since it is equal going to be formed, what will happen? 50% is going to be parental and 50% is going to be recombinants. So that is how we can differentiate between incomplete linkage and independent assortment, right? So in case of incomplete linkage, genes are linked, here genes are not linked. Parental combinations are more than the recombinants, here parental combinations are equal to the recombinants. Equal proportion of gametes are formed in case of independent assortment, but unequal proportion of gametes are formed in incomplete linkage, and it depends upon the percentage of recombinants, okay? Right. Now let's talk about another concept, what we call gene mapping. You know, genes are linearly placed on a chromosome. If they are linearly placed, then we can know the distance between them as well. So genes are separated by a distance proportional to the amount of crossing over or recombination frequency, okay? And the distance are measured in terms of map unit or centimorgan, right? So we can say that map distance is directly proportional to the percentage of recombination or crossing over, right? So let's take one example. Suppose if gene A and B shows 10% recombinance, what it means? It means that gene A and B are 10 map unit apart, and we can show like that. So this is a 10 map unit apart, okay? So this particular concept is utilized in the case of gene mapping, okay? One important thing what you have to keep in mind, recombination frequency between any two genes cannot exceed 40%. So it varies from 0 to 50%. It cannot exceed more than 50%, right? Now, the gene mapping can be of two types, what we call 2-point test and 3-point test. What exactly the word point means? Point means gene. So in case of 2-point test, we can calculate distance between two genes, and that is calculated through the dihybrid cross involving test cross. Now in case of 3-point test, we can calculate distance between 3 genes, but here in addition to the distance, we can also calculate the sequence between genes, okay? Now let's have one question which is usually asked in competitive exams. In a particular plant, gene for tea and red flowers are dominant over dwarf, and when a dihybrid of F1 was test crossed, it produced following progenies. Now we have to calculate distance between T and R, okay? So here what we see, that Test cross is deviating from typical 1-1 ratio, otherwise all have the similar number. Since it is not a similar number, that's why it is easy conclusion that parentals are more, recombinants are less, so it is a case of linkage. Right? So here laws of independent assortment is not operating, so therefore it is a case of linkage. So that means capital T and capital R gene is actually linked one. Okay? So this is the parental one since it is the more number, and rest of the two are the recombinant, this is the less number, right? So gene distance is equal to the number of recombinants by total number of progeny into 100. Now simply you have to put the figure. By putting the figure, what you got? You got 12 map unit or centimorgan, and that is the answer. So this is how such questions are calculated. So only what you have to keep in mind, calculate the number of recombinants, divide by the total number of progeny into 100, that will you will get the gene distance. Right? Again you can see there. So since this number is more, so this is a parental one, this is a recombinant one. So we can easily say that the total number of the progeny is equal to 100. So progeny is equal to 100, recombinants is equal to 18, and the formula will become recombinants by total into 100, right? So it become 18 by 100 into 100. So that's why the 18 become the answer, right? So this is how it is actually calculated. Okay. Have a look at one more problem. In a chromosome, map distance between 4 loci are given as follows, right? Between A and B it is 10, between B and C it is 4, between A and D is 3, between A and C is 6. So what you have to do, you have to calculate distance between C and D. Actually, it is not a question of biology, it is a question of mental ability. So simply what you have to do, you have to draw it and figure it out. Since AC is 6, BC is 4, it is obvious gene C is between A and B. Okay? So gene C definitely is between somewhere, right? So we can show like this. So for gene D there are two possibilities. What are the possibilities? Either it can be of left side of A or it can be of right side of A. So crossover frequency between C and D can be either 9 centimorgan or 3 centimorgan, right? So these are the questions that is usually asked from gene mapping. I hope you have understood it. That's all. Thank you.